This is the model that we used in class today, and I just want to review some concepts of DNA and DNA replication. So on each end, really, there is a 5 end on one and a 3. And what that just means is the sugar and the direction that it is placed in. So if I draw my sugar molecule, basically it's just counting where the carbons are located. So the 5 end basically just means that the direction of the sugar is in this direction, and if it's a 3 end pointing out, that just means the sugar is upside down. So that's where the 5 to 3 comes from. So when DNA is being replicated, what happens first is the enzyme will start splitting our DNA. There are hydrogen bonds between these bases, which it will then split, because remember, enzymes help with facilitating chemical reactions. So I am acting as helicase enzyme. I've now split my DNA. DNA goes and is made copy from 5 to 3. So since 5 is on this end, it's kind of in reverse order for copying. So it copies this way, copy that way. What happens is polymerase helps with adding bases and then also kind of spell checking, making sure the bases are in the correct spot that they should be. Because remember, base pair ruling, C should go with G, and A and T, so it's hard to see, A should also then go with T. So what comes through first is a different enzyme than polymerase. It's just called primase. It's a primer. And it will actually add on RNA, which we'll talk about. I'm not even going to discuss that. And then later be fixed by another enzyme to add all the DNA bases, no RNA. So I have my polymerase coming through, adding on bases. Base pairing, remember, these bases should go together. Notice the difference in the number of pegs. That's just the number of hydrogen bonds. Adenine and thymine only have 2 hydrogen bonds. Cytosine and guanine have the 3. So that's the difference in the pegs that you may have noticed. The DNA will continue to replicate. This strand will also, but it's going to replicate in the reverse order, since it always goes from 5 to 3. And this side is called the lead. The leading side, it just goes right through, quick and easy replication. This, since it has to go this direction, as it unwinds, it will do fragments. But again, you'll have the whole thing together. Sorry for the heat register if that's making a lot of noise. What will then finally occur once I have my DNA strand, our bases are added. Then this is supposed to represent our sugars and phosphates. So now I have one strand. This is my new, this is my old. Same thing here. This is my old and this is my new. And I will have my phosphates and sugars added onto those bases. This is called a semi-conservative, because when they were talking about how DNA is replicated, they were talking about, is it semi-conservative, using one strand to make another and the other strand to make another, or is it completely conservative, which you saw in the animation when it kind of compared the two processes. So that is then, now I have my 2 new strands of DNA. That is how DNA is replicated. Really, the only enzymes that you need to focus on would be helicase unwinds and unzips the DNA, and polymerase helps with adding on. Those are the big enzymes to know about. Thanks. Have a great day.